Finally, I'm going to talk about sequencing. Sequencing begins with PCR and is used to find SNPs and indels. Interestingly, instead of just looking at a single mutation, this technique actually reads each base pair in the region one wishes to sequence. Several sequencing technologies are available. I will talk about first-generation sequencing, which is also called Sanger sequencing. I will also talk about two types of second-generation sequencing. In Sanger or first-generation sequencing, first the DNA is amplified with PCR. The PCR product is then aliquoted into 4 different tubes. Aliquot is just a fancy word that means a portion of the solution, so this means we are going to pipette some of the PCR product in each of these test tubes. In addition to regular PCR reagents, an additional reagent is added, a ddNTP. This stands for dideoxyribonucleic acid. Back in the day, ddNTPs used to be radioactive, but we have better techniques now. And ddNTPs are a special type of nucleotide which stops the extension step of PCR. Each aliquot will receive a different ddNTP. The first step will get a ddATP, the second will get a ddGTP, etc. Then each aliquot is PCR'd again. The addition of ddNTPs by Taq polymerase is random, so sometimes a regular DNTP will be added, and sometimes a ddNTP will be added, which will result in the termination of that extension. In the first example, Taq polymerase might pick up a ddATP after 6 bases and stop there. Another time it extends, it might get a regular DATP at the 6th base pair, but a ddATP at the 8th base pair and stop there. So you can see that you would get fragments all of different sizes. The important thing to note is that fragments in the first tube will end with A. Fragments in the second tube will end with G. Fragments in the third tube will end with C. And fragments in the fourth tube will end with T. After the second PCR, the PCR product is put in a gel, and each aliquot will get its own lane. The gel might look something like this. By reading up the gel, starting at the arrow, you can figure out the sequence of the initial DNA fragment. And that's what you get. After that, you have to write out the complementary base pairs since you are working with complementary DNA. And that's what you get. Today, this is not really done. Instead, each ddNTP is labeled with a fluorescent probe, and the DNA fragments are separated in a capillary tube and read by a machine, like this.